What's up, AP Psychers? Hopefully you are watching this after you've already watched part one of the brain lecture. This is part two. And this part, we are going to talk about some of the special things that our brain can do and some unique, kind of more complicated things, whereas part one was just all about what are the parts and their direct functions. This is a little bit more complicated, but it's really fascinating. And it's fitting that our first heading in this lecture is brain lateralization because it is a type of specialization. To say specialization of the brain, it means that we know certain areas do very specific things. And that's what we're going to talk about here. And one of the first types of specialization is lateralization, meaning what does the left hemisphere do and what does the right hemisphere do? The hemispheres are actually mirror images of each other, but both the left and the right specialize in certain things. The left controls the motor neurons of the right side. So my left brain right now is my left brain is telling my right hand to wave hello. Okay, just like on the right hemisphere will tell my left hand to wave hello. So yes, the left and the right control the motor neurons of the opposite side. The left also controls our ability to speak, so language. It is dominant in our sense of logic and verbal memories. So when we hear language, when we hear something someone tells us, whether it's just take out the garbage to a song lyric to something someone said to us years ago, that tends to be stored in the left side of our brain because that's where language is. The right is more in control of recognizing faces, nonverbal cues like body language or understanding tone of voice when it means a certain emotion, and spatial ability, like for perhaps like I've got my cell phone here, knowing that this is the back of it, that this is, the edge, but that it is still 3D, that it's not just a flat line. Okay. Um, it is a gross exaggeration, not gross icky, like gross large. Exaggeration to say that we are right-brained and left-brained. That is a myth. It is true, however, that we specialize in certain things on the left and the right, but we need both sides of the brain to do pretty much everything. So, for example, let's say we have this idea that the right side is the creative side. Okay, that might be true to an extent because it tends to help us interpret emotions and nonverbal things and music and stuff like that. But imagine that your form of creativity is writing a poem or song lyrics. Well, that's on the left side because that's where language is housed. So to be creative, right side, but yet to write, left side, the both sides really need to work together. And that's true for most things. The right and the left work together. You don't need to write all this down. We've already summarized it on the previous slide, but it just gets into a little bit more specifics, and some people find this pretty interesting. Oh, one more. There you go. Another category that we need to know about the brain is association areas. These are areas of the cortex uncommitted, meaning not devoted to either motor or sensory activity, because it really uses both. The association areas are involved in higher mental functioning, so it will pull from different parts of the brain depending on what the function is. We only need to learn two of them specifically. The first one is Wernicke's area. The second one is Broca's area. Wernicke's, both are on the left frontal lobe and both have to do with language and speech. Wernicke's area is that red dot on my moving graphic. I love these graphics. It's in the left temporal lobe. It is responsible for language comprehension and expression. If you'll remember, the temporal lobe controls the auditory cortex, which means our ability to hear or listen and understand. So left, language, listening, understanding, all in Wernicke's area. Broca's area is also on the left, also has to do with language, but it's a little bit more forward as it is in the frontal lobe. This area is responsible for directing motor movements involved in speech. Remember, our motor cortex is in our frontal lobe, so that section is close to our motor cortex and it helps our mouth move to make speech sounds. So Wernicke's area helps us listen and understand and even helps us decide what we want to say. Broca's area helps the motor of our mouth move to actually make the speech sounds so we can talk. Sometimes both of those areas might be damaged by something called aphasia. Aphasia is the loss of the ability to understand or express speech due to brain damage. We're going to get into this in a lot more detail in class, but please, for the moment, just know that damaging Wernicke's area or damage to Wernicke's area, Wernicke's aphasia, as it's sometimes called, means a person can't understand language. Like they will hear it, but it just won't make sense. Or they'll try to say something and it might not make sense to someone who is listening to it. Damage to the Broca's area or Broca's aphasia means you can't produce language movements. Like in your head, you know what you want to say, you just can't spit it out. Like your brain is damaged and won't let you figure out what word, how to form your mouth to make the word you want to make. Again, we will talk about this in a lot more depth in class, and I'll show you some videos that will help you see what this looks like and sounds like. One thing that makes our brain awesome is that it is very plastic. So what I mean by brain plasticity or neuroplasticity is that our brain has the ability to change or rewire throughout life due to use. So I want you to think about when we talked about the neuron being electrochemical and that the neurons are our brain cells, that we have all these like electric wires going through our head or creating these neural pathways. And the more we do something, let's say learning to tie our shoe, these pathways get stronger and faster and easier to use. And our brain will develop, with every new experience, our brain will add on new dendrites and synapses, synaptic gaps, that will help us do a new function. And the more we do it, so think about when you learn to tie your shoe. At first, it's really hard, but the more you do it, that got easier and faster in your brain, now to the point where you probably don't even really need to think about it. But if we were to totally stop tying our shoes, like completely, we might experience synaptic pruning. And this is that same idea that those dendrites and synaptic gaps that have been formed might kind of break away, because if you don't use those pathways, you will lose those pathways. Sometimes completely, sometimes they just get a little bit slower until you practice that thing again, and then it picks up speed again. The other reason this is really important is that if we have any sort of brain damage, sometimes the brain activity in one area might be able to be transferred to a different location if that first area was damaged. So for example, if you experience aphasia on your left side and struggle to speak, sometimes you can retrain your brain to learn how to speak, but now that might happen in a different part other than Wernicke's or Broca's area. And that connects to our next term called neurogenesis. Neurogenesis is the ability of the brain to generate new neurons or new dendrites and then form new synaptic gaps and things like that, new connections. Scientists have discovered that this ability to generate new neurons and connections is increased by learning, having new experiences, or exercise releasing endorphins. So every time you exercise, when you have physical activity and your body releases endorphins, that helps support the formation of new neurons. But scientists have also discovered it can be decreased, the brain's ability to generate new neurons is decreased by both age and stress. So if someone were to experience brain damage, or even just want to learn something new, this happens most easily in young people and gets harder and harder as you get old. This, in general, neurogenesis is very limited in humans after birth. So it's really important that we protect our brains, because for the most part, if we lose neurons, if we kill off some brain cells, it is not easy to get them back. The next fascinating thing that I would like to tell you about the brain is what happens when we study someone with a split brain. They would have undergone a split brain procedure, probably because they had severe epilepsy and they were having extreme seizures that was actually putting their life at risk. So doctors would perform a surgical procedure where they would cut the corpus callosum, which I forgot to define in lecture 1. So on the first page of your brain lecture, you should have a spot that says corpus callosum. That is the bundle of nerves that makes the left and right hemispheres of your brain talk to each other. In the split brain procedure, those nerves are cut, so you have your left and your right hemisphere still, but they don't communicate. This surgical procedure allows us to study lateralization. In other words, it allows us to study both what the left side does and what the right side does. And what we have found is that hemispheres can actually operate independently from each other and have very specialized functions, as we talked about at the beginning of this lecture. Through split brain research, doctors have discovered that patients visually identify items shown to their right hemisphere, meaning something was shown on the left visual field going to their right brain, and then they could identify that object with pictures, but yet they couldn't say the name of that object. At the same time, if they saw something on their right visual field, it would go to their left hemisphere and then they could say what that item was because that's where language is housed. So while we've already learned about lateralization at the beginning of this lecture, of this part of the lecture, my point is that through these medical procedures that were already done because of a medical need, researchers have been able to learn a great deal more about the brain. And last but not least is mirror neurons. This is not required of you to know this for class. I just think it's fascinating, so I wanted to tell you all about it. In fact, I'm pretty sure I talked about it in class. Mirror neurons are frontal lobe neurons that fire when observing others perform an action. So for example, if you watch someone do jumping jacks, the same areas of your brain light up as if you were doing jumping jacks. And what researchers have determined is that this is an indication of a human's social nature. It allows, these neurons allow us imitation and empathy. In other words, we can copy someone, or just by watching someone, we can start to understand how they might feel doing the thing because our brain is kind of experiencing doing it itself. This is also evidence of observational learning and some proof that we learn by seeing other people do things. So for example, you don't have to write all of these down, but maybe pick one or two. When we see someone get hurt, we might go, "Oh, ow, you just got a paper cut. Oh, I hate that. Yeah, that makes me kind of feel a sense of hurt." When we are trying to get a baby to clap or smile and we go like, "Yay, baby, baby clap," and then they do it back. Sometimes when we watch someone eat something that we think is disgusting, we might go like, "Ew, ew," because we can imagine ourselves eating scrambled eggs. Blech, and they're so gross. Blech. And finally, this is why we often feel happy or sad when watching characters in a movie or TV show or a game show feel happy or sad. Personally, whenever anyone wins big money on like The Price is Right, I get very emotional because I feel like if I was winning that money, I would get very emotional. So, yep, super cool. Mirror neurons. Okay, that's it for the brain. You have learned a lot in these combo 2 lectures, but it's all super important to understanding humans, and it's really important for this class. All right, thanks for listening and taking your notes.